Well, hello everyone. So, what I'd like to do now is take just a little bit of time to review concepts that will be on the multiple choice part of the test. There are questions that look at different patterns of inheritance. What does it mean for a gene to be dominant versus recessive? Codominant, again, with codominant, co means together, so both alleles can be expressed. So, you can wind up with two different proteins being made at one time. And then with incomplete dominance, what that means is in the heterozygous condition, some of the protein is being made, so you get uh, a third phenotype. You don't have like the dominant phenotype, the recessive phenotype. In the case of the heterozygous individual, you get a third phenotype or sort of an in between phenotype. I just think incomplete is in between. So if something is dominant, you're basically making a protein that's going to have a trait. Oftentimes with recessive, you don't make a protein, so you get a different trait. With codominance, you're making two different proteins, so that's sort of its own scenario. And with incomplete dominance, you have one dominant allele, one recessive allele, so you make some of the protein, and you can have, again, a third phenotype. With autosomal versus sex-linked inheritance, think about like a karyotype. When you look at a karyotype, the chromosomes are diploid, right? You have one from biological mom, one from biological dad. So in the case of humans, chromosomes 1 through 22 are all autosomal. So you get two copies of those. So with autosomal patterns of inheritance, we have like big A, little a, or big B, little b, that kind of stuff to represent those alleles. With sex-linked inheritance, the genes are found on the sex chromosomes, typically the X chromosome. And with that, since males and females have different numbers of sex chromosomes or X chromosomes, then you need to keep track of those genes with a superscript on the X chromosome. So you'd be like, XX female with like X big A, and then for males, you could have X little a, Y, whatever the case may be. So with sex-linked inheritance, you have to write in the sex chromosomes, X and Y. And with autosomal, you just can use your big A, little a stuff. All right, the environment can influence gene expression. Genes are instructions for making proteins, right? But depending on what's going on in the environment, it may alter the ability of those genes to produce those proteins or for those proteins to work. So genes are only part of the story. The environment can actually influence the expression of those genes. A nice example is silver maple trees. Silver maples can have like nice, really red leaves in the fall, but that requires an acidic soil. So if the soil is basic, that can change the coloration of the pigments that you see in leaves in the fall. So a lot of times the expression of genes can be influenced by what's going on in the environment. Let's see, crossing over during meiosis one, I'd make sure to review that, so just sort of look at what happens during that time and how portions of non-sister chromatids can be exchanged to increase variation. Independent assortment. What independent assortment means is how one pair of chromosomes lines up has no impact on how another pair of chromosomes lines up during meiosis one. And so because of that, you wind up with different combinations of chromosomes and different gametes, leading to more variation. You'll need to be able to look at pedigrees and figure out, is it dominant, recessive, is it autosomal, or X-linked? Again, there are 4 questions to ask yourself in order. Is it in every generation or does it skip? If it skips, it's probably recessive. If it's in every generation, it's probably dominant. Second question you ask yourself, does a father with the trait pass it on to a son? If that's the case, then it's going to be autosomal, because dads can pass on autosomes to their sons, but dads can't pass on sex-linked or X-linked traits to their sons, because dads pass on Y chromosomes to their sons. So does a dad with the trait pass it on to a son? If yes, it's autosomal. If not, you go to your third question. With the third question, you look at the pedigree and see, are males and females affected approximately equally or unequally? If males and females are affected equally, then it's probably autosomal, and you use your like big A, little a stuff. If males and females are affected disproportionately, then it's likely sex-linked. So you'd use your XY chromosomes to keep track of the gametes or the genes. Okay, I think it's helpful to be able to look at mating scenarios and just sort of in the back of your pocket know what sort of ratios you would expect. So in this first case, if both parents are heterozygous, here there's something autosomal. If both parents are heterozygous, then you would expect a 3-to-1 ratio. You can set up your Punnett square and see something like that. If one parent is heterozygous and the other is homozygous recessive, again, they'll always pass on the recessive allele for one parent, and the other one has a 50-50 chance of passing on the dominant allele. So you get a 1-to-1 ratio. Now, if they ever mention you have a dihybrid cross and that the genes are on separate chromosomes or the genes sort independently, then in a dihybrid cross, that means they're hybrid or heterozygous for 2 characters or 2 traits. And so you would expect a 9-to-3 to 3-to-1 ratio. That's where you did your 16 box Punnett squares. Please never do that. Just know that if it's dihybrid and they sort independently, it's 9-to-3 to 3-to-1. If you want to work out the math, you could just do the calculations on this. Since they sort independently, you can calculate the probabilities independently. So if we look at the A gene, here they're both heterozygous. So what, in terms of having the dominant phenotype, if each parent is big A, little a, then you would have a 3 in 4 chance that the offspring would have the big A character or the big A phenotype. Well, it's same with the B gene here, right? Big B little b times big B little b. You can set up your own Punnett square and see that you have a 3 in 4 chance of having offspring that would have the dominant phenotype. Well, what you can do is say, well, what are the odds they're going to have both? Well, it's 3/4 times 3/4, which is 9/16. What are the odds they'd be recessive for both characters? So if you have this parent with the big A little a and the other parent with the big A little a, the odds of having 2 little a's, it's 1 in 4. Same with the B's. If this parent is big B little b and the other parent is big B little b, the odds of having 2 little b's, 1 in 4. 4th times a 4th, it's 1 in 16. So that's your 1 here. And then with the 3's, the odds of having, if you look at just the A gene, the odds of having the dominant phenotype, again, it's 3 out of 4. But then the odds of having 2 little b's, that's 1 out of 4. So 3/4 times 1/4 is 3/16. So you can sort of see how you can mathematically derive this by calculating independent probabilities. But again, just as a time saver, just know that it'd be 9-to-3 to 3-to-1 if the genes sort independently. Here we got a test cross going on, right? The parent's got the dominant phenotype and the other one's homozygous recessive. If it's the case that one parent is heterozygous for both characters and the other is homozygous recessive, and the genes are on different chromosomes, they sort independently, you would expect equal probability of all those scenarios because, again, the genes sort independently. Again, you can go through and do the modeling. You can do the calculations on your own, but that's what you should wind up with. Now, what if the genes, the A gene and the B gene, are on the same chromosome and they're completely linked? They're always inherited together. If you have one parent's heterozygous and the other is homozygous recessive, then you have equal probability of the offspring looking at one parent or the other. Now, if you get into the Hunt Morgan fruit fly stuff where the genes are linked but that you get crossing over happening, most of the offspring look like one parent or the other, but some are a unique combo. All right. I'd make sure to review what goes on during meiosis 1, meiosis 2. The College Board loves to compare and contrast mitosis, meiosis, so that's a good one. Page 263 in your textbook actually has a figure that walks through that. So I'd suggest taking a look at 263. Non-disjunction. Non-disjunction happens when spindle fibers don't attach to chromosomes, and so chromosomes don't get separated. Let me see if I can find an image with non-disjunction. Oops, it's going to be maybe a chapter. I'm sorry. I should have looked this up ahead of time. Well, for the heck of it, I would just look for examples of when non-disjunction happens. So maybe that'd be like in chapter 15 where they show you something like that. Okay. I'm sorry here. Non-disjunction, it's page 307, figure 15.13. So there you see examples of non-disjunction and what can happen there. Again, page 307, figure 15.13. Meiosis in males versus females. Again, for females, meiosis 1 and meiosis 2 may be separated by decades, whereas males are continuously producing sperm throughout life. So meiosis 1 and meiosis 2 are going to follow in pretty short order. Make sure you can look at a karyotype to make sense of that. Remember, with the karyotype, you have the chromosomes paired up, like the 2 chromosome 1s are paired up, 2 chromosome 2, 2 chromosome 3, and so on. What will be different is the 23rd set of chromosomes. Again, you'd have 2 X's in a female or X and Y in male typically. Let's see. The idea of epistasis. Epi means upon, stasis means standing or staying. So with epistasis, one gene can actually influence the expression of another gene. So you could inherit, dominant, recessive, whatever for one gene, but if another gene is influencing that, you can actually prevent expression of the first pair of genes. So that's a sort of an interesting impact where how genes can actually influence the expression of other genes. Meiosis, how does meiosis create variation? Think independent assortment, crossing over, things of that nature. Let's see, changes in the amount of DNA during meiosis. I think in the lecture for chapter 13, we went through the amount of DNA present in cells at different stages of meiosis. So you might look at that. And then chi-square. If you look at your chi-square POGIL, I think that's a helpful thing to do. Just know how to calculate chi-square. You won't get the chi-square table, like observed, expected, observed minus expected squared, all that business. So just be able to do that on your own. You will get the equation that looks at the summation of all that stuff. So you get the equation from the equation sheet, but you don't get the table. Now you will get the reference table. You will get like what's a significant chi-square value and all that kind of stuff. But going and actually calculating the chi-square is something you'll need to be able to do. All right. As you look through that, if you have any questions, let me know. Otherwise, hope you have a great day. Thank you so much.